Dear students, today I will discuss on T4 bacteriophage. Bacteriophages are viruses that infect and grow inside bacterium. Today I will discuss on importance of T4 bacteriophage, structure and genome of T4 bacteriophage and T4 life cycle. What are viruses? Viruses are obligate parasites that grow inside the host cell. Inside the host cell, it acts as living, and outside the host cell, it acts as a non-living entity. And they are very specific about their hosts. Depending on the host, viruses are classified as animal viruses, plant viruses, bacterial viruses, or bacteriophages. Phage means to eat. Since these bacterial viruses completely destroy the bacterial cell, they are called bacteriophages. Some bacteriophages follow lytic cycle. What is lytic cycle? In this cycle, the virus adsorbs on the bacterial cell surface and injects its genome inside the cell. Then that genome is replicated, transcribed, translated, and the newly formed nucleic acids and proteins assemble to form the new progeny viruses. And these progeny viruses lyse and are released from the host cell. This is called lytic cycle. And some of the viruses, they have a choice between lytic or lysogenic cycle. These viruses are called temperate bacteriophages. In the lysogenic cycle, the viral genome usually gets incorporated into the bacterial chromosome. And at this stage, the viral genome is called a prophage. And this lysogenic state is very stable. And UV light or physical mutagens can excite the viral genome again, and it may come out from the lysogenic state and can follow the lytic cycle again. Now, depending on the nature of the genetic material, bacteriophages are divided into two classes, DNA bacteriophages and RNA bacteriophages. If the DNA is of double-stranded nature, they are called double-stranded DNA bacteriophages or single-stranded DNA bacteriophages. If the genome is made of RNA, they are called RNA bacteriophages. The nature of RNA may be double-stranded or single-stranded. Double-stranded DNA bacteriophages are very important because they are used routinely in many biochemical and genetic research. And few very important double-stranded DNA bacteriophage families are Myoviridae, Cephuviridae, Podoviridae. And viruses of all these families can infect E. coli. Myoviridae got its name from the Greek word myos. Myos means muscle. And the viruses belong to this family have a contractile tail. T2, T4, T6, they belong to this family. The name Cephuviridae came from Greek word siphon or tube. These viruses have a non-contractile tail, but the tail is flexible. And Podoviridae got its name from the Greek word podo means foot. They have a very short non-contractile tail. Now, T-even phages are very important, and mainly T4, because T4 has been used by many scientists from 1940 for research in modern genetics and molecular biology. And T4 system has been used to develop or discover many new findings like mRNA, discovery of mRNA, recognition of nucleotides as genetic material, defining genes by recombinational mutational analysis, etc. And T4 is the first organism where a presence of intron was observed. And how gene splicing occurs, it was observed in T4 only. T4 also produces several enzymes like DNA, RNA ligase, DNA polymerase, and these enzymes are commercially used. So they have a widespread commercial value. T4 ART2 locus. The ART2 locus in the T4 chromosome was used by scientist Seymour Benzer to develop substructures of genes. And many renowned scientists like M. Delbruck, Salvador Luria, Hershey Chase, Alberts, Gisela Mossig, they worked on T4 system. T4 has a very complex structure, complex morphology, and it consists of head, neck, tail, tail fiber, and a base plate. The shape of head is an icosahedron. Icosahedron means it has 12 vertices, 20 faces, and multiple copies of 24 different proteins form this head. 16 of them are involved in the prohead formation, where 5 are responsible for DNA packaging inside the cell. The DNA is packaged inside the icosahedron head in concentric layers, and 3 proteins are required to stabilize the head, and 2 proteins, Hock and Sock, highly antigenic outer capsid protein and small outer capsid proteins, they are also found in the head. The neck separates head from tail or joins head and tail, and the neck is composed of gene products 13, 14.
Ma'am, what is gene product?
Yes, gene product is the protein. Suppose here gene product 13, that means the protein encoded in gene number 13. Now the contractile tail, after neck the tail is present, and the tail is made of 2 concentric tubes. The inner tube or inner cylinder is made of 144 subunits of gene product 19 and arranged in 24 stacked hexameric rings. And outer cylinder, or called sheath, it is also made of 144 subunits of gene product 18, and each stacked ring is 17-degree offset to the right compared to the next ring, and thus it looks, or it takes, an alpha helical right-handed structure. After tail, there is a base plate which is composed of 6 gene products, and gene product 5 and 27 forms a complex and attached at the tip of the base plate, and it has lysozyme-like activity, and this complex, it helps to penetrate the bacterial cell wall. The tail fibers are attached to base plate. 2 types of tail fibers are present, short and long. The short tail fiber is a trimer of gene product 12, and the long tail fiber is composed of 4 gene products 34, 35, 36 and 37. The T4 genome is linear double-stranded DNA, and the length is almost 169 kilobase pairs. And 2 very important features of T4 genome is they are terminally redundant and circularly permuted. Now, what is terminal redundancy? When any DNA contains repeated sequence, similar repeated sequence at both the ends, that genome is called terminally redundant. And if we look into the progeny, each genome carries 1 to 10 genes. Here you see 1 to 10 plus 1, 2, that is repetition at both the ends. This is called terminal redundancy. And if we look into the progeny, each of the genome is terminally redundant, but the starting points vary among the population. This is called circular permutation, because if we can make a circle of each of the genome, then again the order of the genes will be same. This is called circular permutation. Now, the genome has 3 different promoters: early promoters, middle promoters or early late promoters, and late promoters. And early and middle genes mainly encode for proteins and enzymes required for DNA replication as well as for transcription of late genes. And late genes encode for proteins or enzymes required for structure formation, T4 structure formation. And more than 40% of T4 genome is used to carry or to encode proteins needed to construct the T4 structure. And if we look into the genome, then we will see that the genes responsible for each substructure are clustered. Like all the genes responsible for base plate or tail formation, they are clustered here. All the genes responsible for tail fiber, they are clustered here. All the genes responsible for head, they are clustered here. Okay. And the genome is AT rich. That means the T4 genome has high AT content, and it carries a modified base, that is 5' hydroxymethyl cytosine rather than cytosine. And most of the hydroxymethyl cytosines are glucosylated. That means they are attached with the carbohydrate moiety. Now, how the T4 bacteriophage infects its host, that is E. coli. What happens? The tail fibers first attach to the receptors present on the E. coli surface, which are mainly cell wall proteins and lipopolysaccharides. This interaction, electrostatic interaction, attracts the base plate, and the base plate settles down on the cell surface. Now, as I told you before, that there is a complex GP5, GP27, which has a lysozyme-like activity. It lies the cell wall, and the central tube inserts into the cell wall, and the genome, which is present in the head, it will be extruded from the head through tail tube. It will go inside the cell. Now, inside the cell, phage DNA now will multiply. If we look into the timing of the life cycle of T4, then we will see within 2 minutes synthesis of first viral mRNA starts. Within 3 minutes, the bacterial DNA degradation starts. Within 5 minutes, phage DNA synthesis, that is replication of the viral genome, starts. Within 9 minutes, late mRNA synthesis starts. Within 12 minutes, complete head tails appear inside the cell, and at around 15 minutes, you will see first complete bacteriophage inside the E. coli cell. And at around 22 minutes, all the virus progeny will lyse the host cell and will come out from the bacterium. Now, how they synthesize the early proteins? There are 39 early promoters present in the T4 genome. Okay, and these 39 early promoters, viral early promoters, compete with 650 bacterial promoters for about 2000 RNA polymerase of host origin. So, you can imagine it is a tough competition. Then how T4 wins? Because the T4 promoters are stronger compared to the bacterial promoters, and also T4 has some machinery to attract the bacterial transcription machinery. What are they? First, a gene product alt enters into the host with the infecting DNA. That means inside the phage head, apart from DNA, this protein is present, and this protein is an ADP ribosylase. That means this protein transfers an ADP ribosyl group to one of the alpha subunits of RNA polymerase, and this modified RNA polymerase no longer recognizes its own promoters, rather it starts transcription from the viral promoters. Shortly after transcription, two viral enzymes MOD-A and MOD-B are synthesized. These two enzymes are also ADP ribosylase, and they again transfer ADP ribosyl group to both the alpha subunits and completely modify the host cell RNA polymerase. And this modified RNA polymerase now enhances transcription of early promoters, and some of the early promoters are stopped also. And among the early proteins, two endonucleases are very important in T4 life cycle. They are the products of den A gene and den B gene. These two endonucleases cut the bacterial DNA into short double-stranded fragments. Then again, a viral exonuclease cuts the double-stranded DNA into small pieces, or they form the deoxynucleotide monophosphates, and this deoxynucleotide monophosphate pool is recycled to form the viral DNA. There are 30 late early promoters. They are called PM in the T4 genome, and two of the early proteins, ASIA and MOT-A, they modify the RNA polymerase in such a way that now this modified RNA pol, host RNA pol, stimulates transcription of late, early or middle genes. And as I told you before, the T4 genome carries hydroxymethyl cytosine, not cytosine. Okay. So, hydroxymethyl cytosine must be produced from DCMP or deoxycytosine monophosphate. Two viral enzymes convert DCMP to DHMP or DHDP. T4 hydroxymethylase converts deoxycytosine monophosphate to hydroxymethyl monophosphate. Then this hydroxymethyl monophosphate is converted to deoxyhydroxymethyl diphosphate and eventually to hydroxymethyl triphosphate by hydroxymethyl kinase, and then this molecule can be incorporated into the viral DNA. Now, the viral DNA must make sure that it will not enter any cytosine in its genome, because if cytosine enters into the T4 genome, what will happen? It will be susceptible to its own endonucleases that has cleaved the viral DNA. So, to prevent the incorporation of cytosine into T4 DNA, what it does, it converts DCDP to DCMP, because to incorporate any base into the genome, that base must be converted to its triphosphate state. So, the enzyme DCTPase converts DCTP and CTP into CMP. Now, it will not be able to incorporate into the T4 genome. And presence of hydroxymethyl cytosine into the T4 genome creates another problem for the virus. How? There is an endonuclease of E. coli origin which can attack certain sequences containing hydroxymethyl cytosine. So, to prevent that cleavage, the hydroxymethyl cytosine present in the T4 genome is further modified by glucosylation. To viral enzymes, alpha glycosyl transferase and beta glucosyl transferase, they transfer glucose moiety or carbohydrate moiety to the hydroxymethyl group, making it glucosylated. And this glucosylated HMC now protects the T4 DNA from cleavage by bacterium endonuclease. Now, there are 50 late promoters have been found in the T4 genome, and the RNA polymerase is again modified by two viral enzymes, so that now the RNA polymerase only recognizes the late promoters. No more it is required to transcribe from the early or middle promoters. Two proteins, gene product 33 and gene product 55, they modify the RNA polymerase, and now RNA polymerase transcription of the late genes only. And late genes code for T4 head, tail, recombination protein, etc. T4 DNA replication starts just after the immediate early products are formed, and a number of T4 enzymes are required for the T4 genome replication, like T4 DNA polymerase, primase, helicase, recombinase, etc. And T4 DNA replicates in two phases. The first phase of replication begins at multiple origins of replication and proceeds bidirectionally. And as you know that these red arrows are RNA primers. They are required by the DNA polymerase to continue replication in the 5' to 3' direction, and after replication is over, these RNA primers are degraded, and thus the daughter strands produced, they become shorter. Okay, this is the problem with linear genome. Now, if the shortening is not prevented, then after each replication they will lose information. To prevent this shortening, T4 T7 has adopted a mechanism to form concatemers. Okay, and in lambda we have seen that they circularize their genome to prevent this shortening. So, in the second phase of replication, what happens? Two daughter molecules having terminal redundancy, they join together by recombination and form concatemers. But the actual mechanism is not that simple. Okay, I will tell you briefly what happens. The single-stranded DNA end, it invades into the double-stranded DNA having homologous sequence, then initiate and replication is initiated. Then after elongation, branch migration, ligation, they form long concatemers. This model was first proposed by Gisela MOOC, and it is called MOOC model for T4 recombination dependent replication. Another model is there, it was proposed by Formosa and Alberts. This is called bubble migration synthesis. Now, after replication and all the capsid proteins are produced, assembly of genome and proteins, structural proteins takes place. First, the head is formed. The head is formed by more than 10 proteins and scaffolding proteins. Scaffolding proteins help to assemble the head proteins, and scaffolding proteins are removed once the head is formed. Now at the base there is a protein called GP20. It is a portal protein where the tail is attached to the head. This portal protein helps in the DNA packaging. DNA packaging starts with a complex called terminase complex. Terminase complex is made of 4 gene products 16, 17, 17 and 17 prime. This terminase complex first binds to the double-stranded DNA and then it attaches to the GP20 or the portal protein, and then it starts packaging inside the head. And this packaging needs energy. The energy comes from ATP hydrolysis. Now when the head is full, then the terminase complex, which has a nuclease activity also, cuts the DNA from the concatemer irrespective of its sequence. Okay, this mechanism is called head full mechanism. And the DNA present inside the head of T4 is 3% longer than the genome size. That is why it is carrying some extra part. Initial part is repeated at both the ends. That is why terminal redundancy occurs or appears. And suppose from here 2 to 1 is the complete genome length. Since it accepts 3% extra, again repetition of 2, 3. In the next head packaging, it will start from 4. 4 to 3 is the total genome length. Again 3% extra, so 4, 5. This way terminal redundancy and circular permutation will appear in the population. The tail and base plate, they separately assemble. The base plate is constructed of 15 gene products. Tail fibers attached to the base plate, then the central tube is formed on the base plate and the outer cylinder or sheaths appear, and they form the complete tail. Then the tail joins with the head and complete fudge particle is produced. This is a picture where it is shown that inside the E. coli cell complete fudge particles are present, and it has an average burst size 300.
Ma'am, what is burst size?
Yes, burst size is the average number of new virus particles produced inside a bacterium and released from that bacterium. Now, at around 20 minutes of infection at 37 degrees centigrade, almost 300 new T4 fudge particles are released from the bacterium. And for the lysis of the host cell, 2 enzymes are required. They are of viral origin. One is called holin, another one is lysine. And holin is a T gene product. One thing, why these viruses are called T bacteriophages? Because of this T gene present in their genome. The T gene product is a lysine, is a holin protein which always lyses the host cell, and that's why these viruses always follow the lytic cycle and they are called T bacteriophages. So holin and lysine, these 2 enzymes help to lyse the host cell membrane. So the viruses are released into the environment. This is a picture showing lysis of the host cell membrane and release of the new virus progeny. So today we have learned T4 life cycle, starting from adsorption, penetration, transcription of the viral genome, replication of the viral genome, assembly of new virus particles, release of the virus particles from the bacterium. Also importance of T4, how it provides a model system for studying or doing research in biochemical, genetic and molecular biology field. Okay, so T4 is an important tool for biological researchers.